Substructure Analyzer is a user-friendly workflow that performs automatic analysis of multiple fluorescence microscopy images.
It has been developed on the open source software IC and also uses ImageJ functionalities. Importantly, this workflow is affordable without previous knowledge in image analysis.
Multi-channel images are loaded within the workflow and pre-processed in order to improve the signal-to-noise ratio and to remove imaging artifacts. Then, image segmentation isolates regions of interest, also known as ROIs, from the background. Several methods of segmentation are available depending on the level of clustering and the nature of the objects of interest. Segmented objects are saved with a specific descriptor in a specific folder. Fluorescent signals are then analyzed within the ROIs, and multiple features, such as location, size, shape, intensity, texture, spot number, and size, are exported into an automatically created spreadsheet. Download IC from the IC website. Then, download Substructure Analyzer protocol from IC's library of protocols. Open IC and click on Tools in the ribbon menu. Click on Protocols to open the Protocols Editor interface. Click on Load and open the protocol Substructure Analyzer. Protocol loading can take few seconds. The workflow is composed of 13 general blocks, each block working as a pipeline composed of several